Besides carbohydrates, lipids and proteins can also provide respiratory substrates. They can be oxidized in the presence of oxygen to produce molecules of ATP, carbon dioxide, and water. They each have different relative energy levels. Firstly, we're going to look at carbohydrates. The monosaccharide, glucose, is the chief respiratory substrate. Some mammalian cells, for example brain cells and red blood cells, can use only glucose for respiration. Animals and some bacteria store carbohydrate as glycogen, which can be hydrolyzed to glucose for respiration. Plant cells store carbohydrate as starch, and this can also be hydrolyzed to glucose for respiration. Disaccharides can be digested to monosaccharides for respiration. Monosaccharides, such as fructose and galactose, can be changed by isomerase enzymes to glucose for respiration. We're now going to look at lipids. Lipids are important respiratory substrates for a number of types of tissue, including muscle. Triglycerides are hydrolyzed by lipase to glycerol and fatty acids. Glycerol can then be converted to triose phosphate and respired. Fatty acids are long-chain hydrocarbons with a carboxylic acid group. Hence, in each molecule, there are many carbon atoms, many hydrogen atoms, and very few oxygen atoms. These molecules are a source of many protons for oxidative phosphorylation, and so fats produce much more ATP than an equivalent mass of carbohydrates. With the aid of some energy from hydrolysis of one molecule of ATP to AMP, each fatty acid is combined with coenzyme A. The fatty acid CoA complex is transported into the mitochondrial matrix, where it is broken down into two carbon acetyl groups, each attached to CoA. This beta oxidative pathway generates reduced NAD and reduced FAD. The acetyl groups are released from CoA and enter the Krebs cycle by combining with 4-carbon oxaloacetate. For every acetyl group oxidized in the Krebs cycle, 3 molecules of reduced NAD, 1 molecule of reduced FAD, and 1 molecule of ATP by substrate level phosphorylation are made. Finally, we're going to look at proteins. Excess amino acids released after the digestion of proteins are deaminated in the liver. Deamination of one amino acid involves the removal of the amino group and its subsequent conversion to urea that is removed by the kidney. The rest of the amino acid molecule, a keto acid, enters the respiratory pathway as pyruvate, acetyl-CoA, or a Krebs cycle acid such as oxaloacetic acid. During fasting, starvation, or prolonged exercise, when insufficient glucose or lipid are available for respiration, protein from muscle can be hydrolyzed to amino acids, which are then respired. These amino acids can be converted to pyruvate or acetate and enter the Krebs cycle. Finally, we're going to look at the energy values of the different respiratory substrates. We've seen how most of the ATP produced during aerobic respiration is made during oxidative phosphorylation. The greater the availability of proteins for chemiosmosis, the more ATP can be produced. Therefore, the more hydrogen ions that are in a molecule of respiratory substrate, the more ATP that can be generated per molecule of substrate. As the protons, or hydrogen ions, ultimately combine with oxygen atoms to form water. The greater the proportion of hydrogen atoms in a molecule, the more oxygen will be needed for its respiration. The respiratory quotient, or RQ, for different respiratory substances can be calculated using the formula RQ equals carbon dioxide produced divided by oxygen consumed. Because it's a ratio, there are no units. If the RQ value is greater than 1, this indicates that some anaerobic respiration is taking place, because it shows that more carbon dioxide is being produced than oxygen is being consumed.